Hello everyone, welcome to CAPSA. Today I will discuss about terminologies of immunology. So first important terminology is epitope. What is epitope? Epitope is an antigenic determinant which is the smallest unit of immunological reactions. And it can be defined as a small area which is present on the antigen and that is capable of sensitizing T cells and B cells and can react with specific site of T cell receptor or an antibody. It is of two types, sequential epitope, linear epitope, sequential epitope or linear epitope, and conformational epitope or non-sequential epitope. Sequential epitope or linear actually recognized by T cells and conformational or non-sequential recognized by B cells. Now, sequential epitope actually present as a single linear sequence of few amino acids. But conformational or non-sequential epitope actually found on the flexible region of complex antigens having tertiary structure. Now what is paratope? Paratope is an antigen binding site which is a part of variable region or variable domain in an immunoglobulin. So it's a part of V region or domain of Ig or immunoglobulin, or it can be termed as site of an antibody that can react with the corresponding epitope of an antigen. It means a site of an antibody that can react with the epitope of an antigen. Okay. Now, what is haptens? So, haptens are the small molecule after binding to the proteins can elicit an immunoresponse. Proteins means here antibody. So, haptens are of two types, simple hapten, complex hapten. Simple haptens are univalent and non-precipitating in nature, and complex hapten actually multivalent or precipitating in nature. Univalent means it contains only one epitope. But multivalent or polyvalent means it contains more than one epitope. Now, what is precipitating and non-precipitating means? Precipitating means hapten antibody complex which is formed can be visualized by various methods. But non-precipitating means hapten antibody complex cannot be visualized by any kind of method. Now you need to keep in mind that haptens have two properties. Okay, first one is lack of immunogenicity. It means it cannot induce immune response by itself. Only after combined with a larger protein molecule called carrier can elicit an immune response. Okay. Second one is retain antigenicity, or you can say it means immunological reactivity. It means it can bind to their specific antibody or T cells receptor. So that's why they can retain the antigens. These are the two important properties of haptens. Okay. Now, what is adjuvant? So, adjuvant is any kind of substance that can enhances the immunogenicity of an antigen. They are usually added to the vaccines to increase the immunogenicity of the vaccine antigen. Okay. Now, examples are alum, liquid paraffin, silica particles, squalene, etc. You need to keep in mind that lipopolysaccharide or LPS of Bordetella pertussis acts as an excellent adjuvant for diphtheria and tetanus toxoid. Okay, and diphtheria toxin and tetanus toxoid actually act as adjuvant for Haemophilus influenzae type B type of vaccine. Okay, now what is heterophile antigen? So heterophile antigens are the antigen which can share epitopes with each other. Okay. Antibody produced against antigen of one species can react with the other and vice versa. You need to keep in mind that Forceman antigen is a kind of universal heterophil or heterophile antigen, which is present in all animals, plants and bacteria but absent in rabbits. Now, what is superantigens? So, they can act directly on the T cells without an antigen presenting cells or APC. Okay, it means when they activate the T cells directly without being processed by the antigen presenting cell, means directly can bridge between non-specifically MHC2 and T cells. It means it can create a bridge between MHC2 molecule of APCs and T cells. Clear? Now, what are the examples? So, enterotoxin, which is actually obtained from staphylococcus, kind of staphylococcus toxin, and rabies nucleocapsid, that is a viral-like toxin, and Malassezia farfar is a fungal-like toxin. Okay. Now, receptor for these superantigens are variable beta region of T cells receptor. So, receptors are variable beta region of T cell receptor. Okay. So, this is the end of my lecture. If you like my video, please do like, comment, share and subscribe to stay with CAPSA.